Thanks very much indeed, Lenny, and welcome everybody for joining us here at UC Irvine. I'm the director of the Cancer Center here at UC Irvine. We're the only NCI-designated cancer center here in Orange County. We're the fourth oldest one in the state of California. We've had it now for 20 years. So, I was asked by Lenny to explore some of the biology behind ALL, and this is certainly going to be the most biological talk I think you're going to hear at the conference. But I actually was counting quite a bit on Charles to lay the groundwork in the way that only he can. So I'm going to struggle a little bit here without his intro and all this fantastic data, but we're all looking forward to hearing that a little later in the conference. So it's really been his group at St. Jude, and along with many other collaborators from Children's Oncology Group and Pediatric Oncology Group that have really defined the molecular genetic landscape of these precursor B-cell malignancies in a whole series of really seminal papers that relied on genomic analysis of these tumors. The real first observation was that mutations in transcription factors that regulate B-lymphoid development, and these are known predominantly from studies in mouse model systems, are very frequent in B-ALL. And these are genes like PAX5, E2A, EBF1, and Ikaros, which is kzff. In particular, deletions and dominant negative mutations in the Ikaros gene are particularly frequent in pH-positive B-ALL, about 85% of these. Now, we've all known for a long time that pH-positive B-ALL is a bad prognostic category. But it turns out if you parse the genetics, the majority, if not all of the risk segregates with Ikaros, not with the Philadelphia chromosome. So that if you have a Philadelphia chromosome, but you're Ikaros wild type, you have relatively standard risk, whereas if you're Ikaros mutant, but pH negative, you have high risk. And then the last thing that has emerged on the scene has been the definition of pH like B-ALL. This right now has a specific definition that's based upon gene expression profiling. And it's a little controversial, but two papers from Charles's lab, most recently, just 2 weeks ago in the New England Journal, defined this group as having a signature that's similar to B-cerebral positive ALL with activation of stem cell-like programs. And the striking thing about these folks, there are really two striking things about these folks. Number one, they have a bad prognosis. And then secondly, genetically, they have activation of other tyrosine kinases, including sometimes ABL1 itself, and tyrosine kinase signaling pathways, and there's a very interesting story I'm sure we'll hear about from him about rearrangements of the EPO receptor gene and EPO receptor signaling through JAK2 in these leukemias. And then the majority of these also have Ikaros mutations. So to me, as a biologist and a clinician, this raises a couple of questions. Why does Ikaros and how does Ikaros mutation contribute to the pathogenesis of B-ALL? It's very plausible that because Ikaros is required, as I'll show you in a minute, for B-cell differentiation, that this has something to do with the block in differentiation, which is, of course, the characteristic of acute leukemia. But this has never been formally shown experimentally. I think more importantly for clinicians is what is it about Ikaros mutations that confers a bad prognosis? Because as I'll show you in a minute, these folks are all treated with standard chemotherapy, and the remission rate is actually not so much different whether or not you have Ikaros or Philadelphia chromosome. But there's something about them that's causing them to relapse and die at a higher frequency. And lastly, can we use this information to design a better therapeutic strategy? So Mark Litzow just showed you this. This is the ECOG 2993 trial that he spoke about. And I just wanted to point out a couple of things here. One was that in this trial, if you had a PH chromosome, you went on an Abl tyrosine kinase inhibitor. In the early days, it was imatinib. I think currently, most of us would probably be using a second-generation TKI, probably dasatinib. And this has definitely improved the survival of these folks, but it really has not restored their survival to the levels of pH-negative ALL. The second thing is that assessment of minimal residual disease here after the first course and the second course of induction therapy is very important. I want to point out that the complete remission rates in this trial for pH positive and pH negative were not that much different. They were 10% apart. In spite of that, the relapse rates were over twice as high in the pH positive arm. So our ability to obtain a complete remission, or at least a morphological remission, is not what's driving the poor prognosis of these patients. It's really MRD. And this is driven home in this slide here by, this is pediatric data from the Dutch group, where they just looked at the probability of relapse based on minimal residual disease, and they put it in 3 different buckets, low, medium, and high. And you can see particularly in the medium, which is where the majority of the Ikaros mutant patients are, here and here, there's a very striking effect of Ikaros mutation on relapse-free survival. So again, the question is why? Well, what is Ikaros? So Ikaros is a zinc finger transcription factor. It was initially identified almost simultaneously in the labs of Steve Smale at UCLA and Katia Georgopoulos at Mass General. And Katia got to it a little bit before Steve. And so she got to name it, and Katia is from Greece, so she named it and a lot of other family members after Greek mythology figures. So this is Ikaros. There's also Aelos and Hielos in the same group. They're highly homologous. They have a DNA-binding domain and a heterodimerization domain that are both mediated through zinc fingers. Now, the interesting thing about Ikaros is that it does not have strong transactivating properties on its own. It partners predominantly with chromatin-modifying proteins like Mi2 beta to modulate the chromatin of the genes that it binds to and either activate or repress transcription, depending on the gene context. And what Ikaros does in early hematopoietic cells and stem cells is basically inhibit stem cell-like programs, as listed here, and activate programs that are required for lymphoid differentiation. So this is just a schematic of B-cell development. This is all taking place in the bone marrow in mammals. It's a complicated slide, but it shows the different stages of B-cell development as defined by cell surface markers, hardy stages, the rearrangements of their heavy and then their light chain genes, expression of surrogate light chains, intracellular heavy chain, and finally surface immunoglobulin. Now, if you knock Ikaros out in a mouse in the germline, you get a developmental arrest right after the LMPP stage. So no definitive B or T cells are made. Interestingly, if you knock out the other lymphoid transcription factors, say EBF1 or E2A, these cells are arrested at pre-pro-B stage. PAX5 arrests at a pro-B cell stage, so further downstream. So to interrogate what Ikaros is doing in B-cell differentiation, we needed to make a conditional mutant. And Katia did that by placing LOXP sites on either side of Ikaros exon 5 in the mouse. This contains those 2 zinc fingers which mediate DNA binding. So if you activate and recombine this allele, and we use 2 different Cre drivers that express at variable periods during B-cell differentiation, CD2 or CD19 Cre, the results were basically the same. You generated these deleted alleles. These are the 2 wild-type alleles, IK1 and IK2, and here are the ones that are lacking exon 5. These are very similar to IK6, which is a human dominant negative allele that's been discovered in human BALL. It's one of the most frequent alleles found in Ikaros mutant patients. And what I'm going to show you is that if you do this, you have a very profound block in differentiation at the next step down stage, at the large pre-B cell stage. And it's this stage from which high-risk BALL can arise. Okay, so what about the data? So here's the mice. And what you find is that in the Ikaros knockout bone marrow versus the wild type, you see accumulation of these large pre-B cells, which are CD43 positive and BP1 positive, and they cannot downregulate CD43 or BP1. Similarly, they cannot upregulate CD2 or IgM, as shown here. Now, these cells are polyclonal by immunoglobulin heavy chain gene rearrangement, and they have germline light chain genes, so they're pre-B cells. The block in differentiation is not rescued by providing them with a rearranged kappa transgene. So in this case, we crossed in a transgene for a rearranged kappa gene, and you can see that despite expression of this kappa gene here, the cells are still arrested at the pre-B cell stage. So there's another step that's required besides simply immunoglobulin gene rearrangement. And amazingly, these cells fill up the entire B cell niche in the bone marrow. You can see that the total number of B cells in wild-type or mutant bone marrow is the same. And they're all composed of these pre-B cells, as defined by these markers here. But they don't go outside the niche. They don't interfere with myeloid or erythroid function. The hematocrits, platelet counts, and neutrophil counts in these mice are perfectly normal. They don't circulate to any extent in the blood, and they don't populate peripheral lymphoid organs like the spleen or the lymph nodes. The mice are basically, besides from being B-cell immunodeficient, they're perfectly healthy. Now, if you profile these cells, you find that they have very high activation of integrin receptors and activated integrins and downstream signaling pathways. And I think this is very interesting because this is all epigenetic. It has to do with the lack of occupancy of canonical Ikaros binding sites in the genome and the repopulation of different promoter complexes on other genes. So this is missed by the genomic analysis. You don't see this because there's no mutations in any of these genes or these pathways. These cells express high levels of activated beta-1 integrins by antibody, and they show a very highly adhesive phenotype to stroma in vitro. You can just see here, they form these very dark areas, almost reminiscent of cobblestone area forming cells. And if you do confocal microscopy, you can see where the nuclei are in blue and the stroma are in green, that the leukemic cells here have very high activation downstream of the integrins of FAK, focal adhesion kinase, and that's just shown here in the red. And this is the corresponding wild-type bone marrow here. And you can make extracts and do Western blot, and you can see the same thing, very prominent activation of focal adhesion kinase. Now, the non-adherent cells in these populations really have the properties of differentiated cells. They've differentiated down the pathway to small pre-B cells. They have activation of P38 kinase signaling, and they have prominent calcium fluxing in response to ionophores. So these are both parts of the B-cell receptor signaling pathway that are characteristic of differentiated B cells. By contrast, the pre-B cells that are arrested in the mutant Ikaros mice are very proliferative, but they're absolutely dependent on stroma for this. You can see the proliferation profiles here. If you look at their ability to survive off of stroma, you can see that in the absence of stroma, the mutant cells undergo apoptosis at high levels. In the presence of stroma, they can survive even the absence of cytokines such as IL-7. So they're really stromal dependent. And if you look at their signaling, they have highly activated proliferative signaling, particularly in the ERK pathway, and also with activation of cyclin D2. So as I mentioned, these cells are not, I think this slide is out of order. Yeah, sorry. These cells are not by themselves leukemogenic. I'm sorry, no, it wasn't out of order. These cells are not leukemogenic, but you can have them progress to acute B-cell leukemia if you serially transplant them into recipient mice, and that's just shown here. If you take the wild-type pre-B cells, they don't cause any disease, but the pre-B cells derived from bone marrow of these primary mice, when transplanted into secondary mice at very long latencies, these are months now, will generate acute B-lymphoblastic leukemias that will kill the mouse. These are highly invasive tumors. In particular, they completely repopulate the bone marrow. And the cause of death, at least in these mice from the CD19 Cree arm, is actually bone marrow failure. They're dying of anemia from lack of myeloid and erythroid function. Interestingly, if you look at the tumor cells in these mice and look at their immunoglobulin heavy chain gene rearrangements, in this case it's done by southern blotting, they're oligoclonal or monoclonal. So to a mouse geneticist, this infers that there's additional events that have taken place to allow them to become fully leukemic. Well, what might those events be? Well, one very obvious possibility, again, based on the observation of activation of tyrosine kinases in pH-like leukemia, is that this is activation of another tyrosine kinase. So to directly test this hypothesis, we took a BCR-ABL1 retrovirus and introduced it into these pre-B cell, pre-malignant pre-B cells from either Ikaros dominant negative or wild-type mice, and then transplanted these into syngeneic recipient mice. We'd shown years ago that if you do this with wild-type bone marrow, you can generate mice that have acute B-cell lymphoblastic leukemia that looks very much like pH-positive ALL in humans. And the question was, what does Ikaros mutation do to this? And the answer is that it greatly, greatly accelerates the leukemogenic process. So here are the survival curves for mice that receive these cells. You can see that all mice that receive wild-type cells die, and they die by 7 weeks. And with heterozygous, or more prominently with homozygous mutations in the dominant negative Ikaros, you see just an incredible acceleration of disease. These mice are dying by day 14. They're sublethally irradiated. That's about as fast as a mouse can die in this assay. And they're dying of bone marrow failure. They have basically no erythroid cells left. Now, one hint to this acceleration is provided by interrogating the number of leukemia-initiating cells in this assay. So the retrovirus is valuable because it is replication defective. It will land in a chromosome, and then it will not move. So it serves as a unique clonal marker for that cell and its progeny. And you can interrogate this by simply preparing genomic DNA from these tumors and doing a southern blot here with the appropriate probe. So you can see that the number of clones that initiate the leukemias in the wild-type bone marrow is oligoclonal. It's 3 to 5 clones. This goes up about 3-fold in the heterozygotes, and it turns into a smear in the homozygous. If you do limiting dilution on this, there's approximately a 300-fold increase in the leukemia-initiating or leukemia stem cell frequency in these mice. To me, that confirms one of our hypotheses, is that one of the things that Ikaros deficiency is doing is increasing the likelihood of generating a leukemic stem cell, probably through its block and differentiation, although we need to prove that. You can see that, interestingly, these mice express the dominant negative alleles, and the wild-type leukemias that come up have not rearranged Ikaros. They've arrived at their differentiation block through another pathway. And then lastly, these leukemic cells that come out of these mice share the same adhesive phenotype, in this case to fibronectin, but also to stroma, that the pre-malignant cells do. If you look at these cells and expose them, because they express BCR-ABL, expose them to ABL kinase inhibitors like imatinib or dasatinib, you can kill them. But there's a modest but very reproducible increase in the IC50 when they're grown on stroma. And again, when they're on stroma, they're very highly adhesive. And if we use a FAK inhibitor, in this case, this is a commercially available FAK inhibitor, you can see that you can reverse the phospho-FAK profile of these cells, both the pre-leukemic and leukemic cells, in vitro. You can block their adhesion to stroma, and you can induce apoptosis selectively in the Ikaros mutant cells and not in the wild-type cells. And you can actually do this in vivo to a certain extent. These are published data actually from pre-leukemic mice. These are the donor mice for the transplant experiment. And you can see that you can dramatically and significantly reduce the number of pre-B cells and increase apoptosis in the bone marrow of these mice with just a few doses of a FAK inhibitor. So we believe that this suggests a new model which needs to be tested for high-risk BALL. So wild-type pre-B cells have this transient phase where they're adherent to stroma as large pre-B cells, and they activate FAK, and they have activation of their survival and proliferation pathways, but they very rapidly differentiate into small pre-B cells that have downregulated their integrin function, and these have switched on differentiation pathways, including rearrangement of their light chain genes. By contrast, the Ikaros mutant cells are locked in this adhesive large pre-B cell state. And the idea would be that with secondary tyrosine kinase activation, and I see Charles in the back now, he's entered the house, I'm sure you'll hear from him about more of these events, that these types of signaling events can convert them into a leukemic cell that is then able to circulate and cause all the problems that we know about in leukemia. But I would posit that it may be this cell, the one that's in the bone marrow and highly adhesive, that may be responsible for the minimal residual disease and our inability to cure these patients. So what is it about Ikaros in this? Well, I would suggest that perhaps these other transcription factors are arresting B cells at stages that are prior to this adhesive phenotype. And it may be only the Ikaros, or predominantly the Ikaros mutant ones, that have this adhesive phenotype that might be translated into increased minimal residual disease and the inferior prognosis. Again, this is a hypothesis. It's unlikely to be exactly true, but I think there's enough evidence there to make it worth pursuing. So our next steps here are we're testing the efficacy of FAK inhibitors plus or minus ABL kinase inhibitors in these preclinical models. And we're moving forward to open a phase 1 clinical trial of FAK inhibitor in this disease. Fortunately, there are a couple of FAK inhibitors that are in clinical trials right now from GSK, Novartis, and Verastem. They're mainly being used in solid tumors, particularly in mesothelioma. So we're going to try to move this, at least as a phase 1 dose escalation trial, into the clinic quickly. And then ideally to try to move to a phase 1B or 2 trial where we target it more to high-risk BALL. So I think I'm on time, Lenny. This is just the laboratory that I have, a very small lab that has done the majority of this work. I'd like to thank Nilu Gina, who's in the audience, who did a lot of this work. And then our collaborators, Katya Georgopoulos at Mass General and Elizabeth Paeta, who Mark already mentioned at the Leukemia Bank for ECOG. And before we all come up and take questions, we have UC Irvine and Tufts, my former institution, for a number of years has been sponsoring a reception late night at ASH We're going to do it again this year. So I'd like to invite anybody in the audience who's attending ASH to join us on Saturday night. No, it starts at 9 p.m. We try to catch people after their ad board or dinner or whatever, but it's a good time. It's an excellent time for networking and to relax a bit and see your colleagues. So please join us. Thank you.